The male, and then as we'll see there also, of course, produce hormones that not only drive that process of gamete production, but also have other effects on the body, right? Of course, the effects on bone density, muscle mass, and so forth. Okay? Now, the male gamete production and hormone production is really the sole purpose of the reproductive system. And the female, of course, as eutherian mammals, mammals with placentas that carry their young inside, of course, the female has the additional burden of carrying the young in through the development of gestation phase. All right. So we'll take the simple one first. We'll do the more complicated one next week. All right, so what do we mean by producing a gamete? Well, as you know, all of your cells have 46 chromosomes, 46 strands of DNA. Now, those 46 strands of DNA represent 2 complete copies of your genome. Right? Each of your cells has 2 copies of your genome. Each one of those is in 23 chromosomes. So what we have are 23 pairs of homologous chromosomes. Homologous means they're similar but not exactly alike, right? So I have chromosome 1 for mom, chromosome 1 for dad, chromosome 2 for mom, chromosome 2 for dad, all the way through chromosome 22. 1 through 22, those are so-called somatic chromosomes. The 23rd pair are the sex chromosomes. As a male, I have an X and a Y, and the females have 2 X's. Okay? Now, chromosome 1 for mom and chromosome 1 for dad are very similar in their sequence of DNA, but there are genes that have some differences in them, some variation. Right? We have different forms of the genes on each of those, and we'll talk more about that as we get into things like blood typing. What you want to do to produce a gamete or sex cell is to produce a cell with only 1 copy of the genome, only 23 strands of DNA, 23 chromosomes. So that when it's then combined with the gamete from the other sex, you produce a cell with, once again, 2 copies of the genome. Now, the process of producing these gametes begins in the same way as the process of normal cell reproduction, what we call mitosis or somatic cell reproduction. We first begin with interphase, right? And you recall in interphase, one part of that was called S phase, and in S phase you duplicated all your DNA. So that you ended up with 92 strands consisting of 46, each one attached to a copy of itself. All right? And then you would split those apart, those were called the chromatids, and then you would split them apart to produce, once again, 2 cells with 46 strands or 23 pairs. The only difference now in this process called meiosis is we're not going to divide once, we're going to divide twice. The first division, we're going to separate the homologous chromosomes. So after interphase and S phase, each of your chromosomes in their condensed form looks like that. There's 2 copies, 2 chromatids attached to each other. You may recall that structure where they attach is called the centromere. Okay, so this will be, let's say, chromosome 1 for mom, chromosome, that's 1 sub M, and this is 1 sub D, right? Chromosome 1 for mom, chromosome 1 for dad. That's what those look like. So that first division, we're going to split these apart. That first division is the separation of the homologous chromosomes. This is very different from what happened in mitosis. In mitosis, we didn't care which chromosome it was, as long as they lined up in the middle at metaphase and then split apart in anaphase. With meiosis, we have to do something different. We have to separate those homologous chromosomes. So So that one goes from one, dad goes one way, and one from mom goes the other. Okay? Now there's no reason to say that all the dads have to go this way and all the moms have to go that way. You can mix and match them per chromosome. But you do have to separate the homologous chromosomes. Okay? So that's what happens in meiosis 1, the first division. The homologous chromosomes line up. It's called synapsis, meaning they come close together. That's what synapse means. And then in that first division, the homologous chromosomes separate. Now we're left with 2 cells that each have 23 chromosomes, or only 1 copy of the genome, which is half the normal number, right? We are diploid organisms. Diploid, meaning our normal state is to have 2 copies of our genetic code. There are other animals that have many more than that, polyploids. Right, we're diploid. When we get down to 1 copy, we have half the normal number, so we call this haploid. Not monoploid. Monoploid is the word we use for organisms that normally only have 1 copy, right? These cells are haploid. They have half the normal number. So once we're finished with meiosis 1, now we have a couple of haploid cells, but those still have 2 copies of that chromosome stuck to each other, still 2 chromatids. So the second division is just like mitosis. We're going to line them up in the middle and split the chromatids apart so that we end up finally with up to 4 cells that have each 1 copy of the 23 chromosome. And that is our gamete, barring morphological changes. Right? So that cell can then be combined with another cell from the other gender, the other sex. I use the word sex in biology, right? The other sex, and then we end up with a cell with 46, meaning 2 copies of the genome. All right, so that's what we're doing. We're splitting twice. That first division, called meiosis 1, we're separating homologous chromosomes. Second division, meiosis 2, is very similar to mitosis. We're splitting sister chromatids. Now, it's an advantage to an organism when it reproduces to maximize the genetic diversity of its offspring. Right? We have evolved a number of mechanisms to maximize the genetic diversity of our offspring. Because like our country has benefited so much from our cultural diversity, the offspring benefit from, or the survival of the species benefits from, genetic diversity. Because genetic diversity means the more diverse the population is genetically, the more likely there are to be individuals who can survive future unknown challenges, like a new disease or something like that. Okay? When you end up with small populations that interbreed and diminish genetic diversity, the sort of common trope is that they end up with mutations. So what happens there is genetic mutations aren't filtered out, so they're concentrated into a small family. But also it means that the species is less likely to survive future events, right? A species like the panda or something like that has very low genetic diversity is really only going to survive through human intervention, right? If we ever stop paying attention, not only because of our habitat destruction, but simply because of random events in the future, some species like that with low populations and low genetic diversity is just doomed. Right? Humans don't have that problem. Right? There's 7 billion of us. We did at one point go through something like that. The evidence in our genes says that we went through what's known as a genetic bottleneck, where the population dropped very low, probably around 100,000 years ago. We think it was caused by the eruption of a supervolcano in what is now Lake Toba in Indonesia. That this caused a global climate change that may have reduced the population of humans down to thousands, a few thousand. We don't know for sure, but there's evidence in our genetic code that that's what happened. But now we don't have that problem, right? There's plenty of us, but we still have these genetic mechanisms, okay? We still have these mechanisms to maximize diversity. So the first obvious way to do it is by simply mixing and matching chromosomes. You're going to produce gametes, right? They'll split apart in that first division where they separate homologous chromosomes. Like I said, there's no law that says all of dad has to go one way and all of mom has to go the other. You can send mom, 1 from mom, 2 from dad, 3 and 4 from mom, right? It's one way and then the opposite the other way. It's random. Okay? So that's one way to do that. The other way is by mixing and matching chromosomes. This happens during prophase of meiosis 1, right? The homologous chromosomes come together. Let's say this is chromosome 1. The yellow is mom and the red is dad. Doesn't really matter, okay? Chromosome comes together. Synapsis. Well, what happens is these two chromatids that are touching each other will cross over. And then part of the chromosome from one will detach and go to the other. So now we've produced a unique chromosome here, a chromatid, which will become a chromosome once it splits, that is now a combination of mom and dad. This didn't exist before. This is a unique combination. So some of the potential gametes will get these blended chromosomes. And where that happens and the degree to which it happens is also somewhat random. And so we can produce tremendous combinations of our existing genes in our offspring. So if you are one of multiple siblings, or if you have multiple children, you know, of course, that they can be very, very different, not only in their appearance, but their behavior. And of course, this is one reason for that. Okay, so we'll spend some more time talking about this process in lecture and the end result. But for now, I want to move on to talking a little bit about it in the context of the male and female reproductive system, and then we'll move on to talking about the anatomy of the male. So both in the male and the female, we have a gamete-producing structure. In the male, this is called the seminiferous tubule. I can't remember if that is a it's not. Seminiferous tubule. These are tubes. There are a great many of them in the testes, and in the wall of these tubes, we produce the gametes. Of course, in the male, there is an additional process of morphological change, right? Meiosis produces basically a spherical cell. But of course, the male gamete is spermatozoa. It has a head and a tail, a flagellum. So there's an additional process that goes through. The entire process of making male gametes is called spermatogenesis. And that consists of meiosis, to make the genetic part of the gamete, and then spermiogenesis. Spermiogenesis is the morphological change, the shape change between being a sphere and being a tiny cell with a head and a flagellum. Okay? Genetically, it doesn't change going from a sphere to the sperm, right? It's a shape change. In the female, you've got oogenesis. This is the only place where you'll find words beginning with two O's. Oogenesis is the female. And this relies only on meiosis, because the female gamete is a sphere. It's a very large spherical cell. It's in fact the largest cell the body produces. Either body produces. It's, if you've got good vision, it's just barely visible to the naked eye. Right? So in the male, the process occurs in the wall of the seminiferous tubule. In the female, the process occurs in the follicle, which is located in the cortex, or the outer part of the ovary. So in the ovary of the female, the follicle produces the gamete. In the male, it's the seminiferous tubule within the testes. Right? The testes and the ovary are referred to as the gonads. Gonads are the gamete-producing structures. Okay. So here you can see the wall of the seminiferous tubule. We'll look at a slide of this in a moment, or in a little bit. This is the basement membrane that surrounds the wall. You can see it there. And what's going to happen is stem cells are going to be reproduced here just beneath the wall, or just beneath the basement membrane that surrounds this tube. These are the stem cells for producing gametes. Those are sometimes called germ cells. In the male, they're called spermatogonia. Spermatogonium would be the singular. In the female, they're called oogonia. Oogonium would be the singular. Now, as we'll learn next time we get into the female gamete production, no female in this room has any oogonia. But the males have plenty of spermatogonia, right? This is because of a very, very different strategy that the female and the males use to produce gametes. The females, oogonia, all begin meiosis and become what we call oocytes before she's even born. This gives her a limited number of potential gametes that she produces throughout her life. Males maintain those germ cells throughout their lives and produce countless trillions of potential gametes throughout their life. The female, as I mentioned before, and as of course you well know, has a much greater personal physical investment in producing the young. Not only, of course, does she perhaps want to reproduce. I'm not talking about the individual desires of the person here. I'm talking about this from a biological context, to protect and nurture the young, but she also doesn't want to die, right? Because of course it's very risky and it takes a lot out of her to produce an offspring. The male, of course, also wants to spread his genes around and has very little in terms of personal risk to fathering a child, right? Now, of course, being responsible and wanting also to support his young, he will, of course, invest a great deal in that normally. But in terms of the physical risk of the process, there's not much there, okay? So the male strategy is to produce many millions, and then throughout the life, trillions of gametes to maximize the probability that he will fertilize an oocyte and father young. The female, on the other hand, though she may, from a biological point of view, wants to be the mother of young has a great risk here. And so she makes it just barely possible every now and then throughout her life for her to become pregnant. Okay? One is a very, very, very conservative strategy. The other one is a much less conservative strategy. Obviously it works because there's 7 billion of us, right? But in terms of personal strategy, right? Because, women, my great-great grandmother died at age 50 after having 9 kids, right? Takes it out of you. Now, I'm not saying that everyone who has 9 or 10 kids is not going to live a long time, but of course, childbirth was a lot riskier. Medical science wasn't where it is now, but okay, it wears on the body. So we have, again, 2 very different strategies there. Okay. Again, this is happening in the wall of the seminiferous tubule, but just beneath the basement membrane, you have the spermatogonia. And these will then divide. And they will divide and replace themselves. So there's your basement membrane. So the first thing that happens is they divide and replace themselves, right? That's the part the female doesn't do. Then one of those 2 daughter cells, even though this is a male, we still call them daughter cells, because they could become a daughter, part of a daughter. All right? 2 daughter cells are produced. One remains behind as a stem cell. The other one differentiates by entering meiosis. Once it enters meiosis, it becomes known as a primary spermatocyte. Right? It then completes meiosis 1, entering meiosis 2, where it's then known as a secondary spermatocyte. Finally, completing meiosis 2, it is known as a spermatid. Now you'll notice in the process, we change some designation. 2N means diploid. N means haploid. So again, once we divide those homologous chromosomes, we split those apart, we are technically haploid, even though they still have 2 chromatids to each chromosome. Completing meiosis 2 splits the chromatids, so now there's only 1 chromosome in each of these. Also, a little bridge of cytoplasm. So meiosis 1, meiosis 2 complete, but cytokinesis, the process of dividing the cell into 2, is not quite completed on these until later. There remains a small little bridge of cytoplasm between them. And that's because half of these cells have something that the other half don't have. I guess what that is? This is a male, remember? We divide the homologous chromosomes. That means half of them have something the other half don't have.
X and Y?
Right? Either an X or a Y, right? Half of them have an X, half of them have a Y. What matters is the X. The X chromosome is much larger than the Y. The Y, there's this argument that males are becoming extinct or genetically unnecessary because the evidence shows that throughout human evolution, the Y chromosome gets smaller and smaller and smaller with fewer and fewer genes. Okay, it's the whole reason behind sexual reproduction, behind having 2 sexes and then bringing them together, is to maximize the diversity of genetic offspring. If you could have 2 separate females that somehow could come together and produce a gamete, I guess you could do that in theory in a lab, right? Then there wouldn't be a need for a male. But having 2 sexes is a convenient way to divide up these sets of genes that can then come in together, right? That's what evolution has given us. All right, but half of them have an X, the other one don't. Now the X chromosome has genes on it that both of them will need to complete this process, right? You're not viable without an X chromosome. And so that's when we have to have that cytoplasmic bridge. So those, whatever proteins, I don't know what they are, but those proteins that go between, from the X chromosome to the cells that only have a Y, have a mechanism to do so. All right, lastly, we have spermiogenesis. Now again, no genetic change. We changed the spherical spermatid to the familiar spermatozoan or sperm cell, which at this point is still not viable. It has to go through an additional maturation process. Like many men, sperm take a long time to mature. Sorry, I'm allowed to make that joke because I know. But this is the physical change that has to occur here. Now again, this happens in the wall of the seminiferous tubule, and as it happens, the prospective gamete moves from the superficial part just beneath the membrane, just beneath the basement membrane, all the way to the lumen. And so here you can see in the lumen, you have this, you can see the tails of the sperm sticking out there. Now you don't just have the spermatocytes in the wall of the seminiferous tubule. There's another cell called a Sertoli cell. Also called a nurse cell. That's what I learned in high school. A nurse cell. And its new name, right, because I've told you several times that there's this movement to remove all the old dead white guys' names from these anatomical structures. So they want to get rid of Sertoli's name. So what they came up with was sustanacular cell. Right? It sustains and nourishes the sperm. Okay, nurse cell, I guess, was too colloquial, so they called it a sustanacular cell. But I like Sertoli is easy to remember. All right, so these are the cells you see here stretching from the superficial part to the lumen. And they do a couple of things. Again, they nourish the cells as they go along, they protect them. They play a big role in spermiogenesis. They're part of how we remove that extra cytoplasm and compact down the sperm. But then one more thing they do here is they join together just beneath the layer where the spermatogonia are, just beneath this part. They'll join together with tight junctions. And this forms what we know as the blood-testis barrier. Now remember, the blood-brain barrier is that there's things in the blood that you don't want floating around in the cerebrospinal fluid, like glutamate. So we have to have this barrier. The blood-testis barrier is an immune barrier. There's a high probability that the immune system will attack the sperm once they enter meiosis. And the reason is that crossing over, right? We have the two chromatids cross over. There's a possibility, it's probably individually very low, but if you do this a few billion times, you're going to end up having this happen quite often, is when you cross over here, you modify a gene in some way that it makes a protein that looks like non-self to the immune system. And so we isolate these cells once they move intrameiosis 1 and do that crossing over, they're now isolated from the immune system. Right? So again, they're going to be destined to be expelled from the body or reabsorbed if they're not used. Okay? Now, you'll notice that in this process, 4 sperm are produced from each from each spermatogonium that entered this process. Again, very liberal strategy here. Make as many gametes as you can. Female, much more conservative. Right? The male constantly produces sperm all the time. Now, the rate at which they're produced varies throughout life. Peaks in like the late 20s, early 30s, and then declines throughout life as testosterone levels drop. You don't really see the results of this until quite late in life. You see the loss of muscle mass and the other changes you see in the male body as they reach that age. It's called andropause. So there is a male menopause, andropause. It's just very slow, right? The female is much more abrupt. So you see the symptoms are much more obvious. All right. So constantly. And again, I wouldn't want to speculate on how many total sperm the average male will produce in his lifetime. All right. The female Of course, they don't start producing them until they reach sexual maturity. Same thing is true of the female. She doesn't start producing potential gametes until she reaches sexual maturity. And then she only produces one a month on average that is not even a gamete yet. It has the possibility of becoming a gamete. Right? So we see that process here. Again, this part, or oogonia, before the young lady is even born, all of the oogonia enter meiosis 1. And then they stop and they hang out. There's millions of them when she's born, but they just kind of hang out over the next, however many it is, years, till she starts her periods, till she starts her potential reproductive life. Menarche it's called. Hopefully not reproducing at that moment, but historically, that possibility would have been more common. And over that time, most of those will die off. They'll just be reabsorbed. And so she has maybe a couple 100,000 when she starts her menstrual cycle. Okay? And then on average, one a month will be ovulated. Now, that is driven by the monthly rising and falling of hormones, right? Again, we'll talk about this hormonal control. It's much more complicated, once again, in the female than the male. Though the same control mechanisms are used by both. Driven by that monthly cycle of hormones, a few of those oocytes that have stopped will now start again, and on average every month, one will reach meiosis 2 and be ovulated as a secondary oocyte. It's not until and if it is fertilized that it encounters a sperm that will complete meiosis 2 and produce an ovum, the final gamete. So female gametes don't even exist until sperm are already inside of them. Again, very, very conservative strategy. Now you'll notice she's not making 4 of these, she's only making 1. The other half of the genetic material is thrown away. It's squeezed off to the side to one pole of this giant cell to be known as a polar body. That first polar body sometimes divides in meiosis 2 as well, and you'll get up to 3 polar bodies in one final ovum, which exists for less than 24 hours before the nucleus of the sperm, which has already entered it, fuses with it to produce a zygote. Again, very conservative strategy. And we'll talk about the anatomy of that next time. I want to now focus on the male reproductive anatomy. And we'll start with the scrotum, which contains and houses the gonads, right? The male gonads cannot produce viable sperm at core body temperature, and so the scrotum allows the testes to be held at a temperature lower than core body temperature. They're covered, of course, with skin. Along the way, as I discuss this, I'm going to talk about homologous structures. So we won't get too much into the development of the male and female genitalia. But of course, it's of interest, but there are many ways in which it kind of deviates a little bit, right? So you have sort of intermediate sexes or people with slightly different anatomies that tend towards more one traditional sex or the other. So I'll talk about that homology. So there's skin that's got hair follicles in it covering the testes, and this is homologous to the skin of the labia majora in the female. Now, one thing that's unique about this skin is instead of having just little bits of smooth muscle associated with each hair follicle, there's a sheet of smooth muscle beneath the skin of the scrotum. This is known as the dartos muscle or the tunica dartos. And what it does is it causes that skin to wrinkle, reducing its surface area or its effective surface area and reducing heat loss. Right, so it's part of a thermoregulation mechanism. Now there's another muscle involved. This is a skeletal muscle. The inferior fibers of the internal oblique actually descend into the scrotum. Now, unlike the other internal oblique fibers, these are not under voluntary control, but this is then known as the cremaster muscle, and this will raise and lower the scrotum, moving it closer to the body and further away. Again, this is a thermoregulation mechanism. I remember when laptops first came out, people actually put them on their laps. And of course, people don't do that anymore because they're so darn hot. But they weren't quite as hot then, but they were warm. And there was thought that some men were suffering lower sperm counts because of working in that way. Because at higher temperatures, average temperatures of the testes leads to a lower sperm count. Now, of course, the testes again are housed in the scrotum, and there's a set of vessels and structures that tie them into the core of the body. They actually develop inside the abdomen, and you'll see this when we look at the vasculature. And then near the time of birth, they descend through the inguinal canal, which is the canal between the body's core and the scrotum. They descend into the scrotum. Sometimes they don't. It's a condition called cryptorchidism, crypt meaning hidden, orchid meaning testis. And in that case, the testosterone will be produced. There's nothing stopping that. But the little boy, if that's not corrected before he reaches sexual maturity, will be unable to produce viable sperm. Right? He will also have a tremendously increased risk of testicular cancer. Okay? So there seems to be a correlation between average testicular temperature and testicular cancer risk, which is why I thought, that with all these laptops, there's going to be a spike in testicular cancer, but I think technology took care of that because the darn things got so hot, nobody would actually put them on their laps. All right? But that is a condition you may have heard of. Okay? So I've mentioned the skin, the dartus muscle. I don't think there's really any homology in the female to the dartus muscle or the cremaster. And then we've got some fascia. Okay? And then there's a layers surrounding the testis called the tunica vaginalis. And this is actually part of the peritoneum. Remember, the peritoneum is the serous membrane that surrounds all the visceral organs. And some structures, like the genitals and the reproductive organs, are only in contact with the parietal peritoneum, the outer part, which they're called retroperitoneal, behind it, but really in contact, just because there's simply none of them in the front. They're called retroperitoneal. Okay? So we've got this parietal peritoneum. So let's say this is the testis. What it does, so there's your parietal peritoneum. What it does is it turns and it wraps itself around the testis like this and then goes back. And that creates a little fluid-filled space here. Okay, and sometimes you'll get an accumulation of fluid in there. Testicular edema surrounding that. All right, and then finally, on the very outer surface of the testis, you have a thick connective tissue layer. It's white, so it's called the tunica albuginea, alba meaning white. Okay. Now again, this testis is served by a series of vessels and structures that pass through the inguinal canal. The inguinal canal is a fold in the aponeurosis of the internal oblique that allows that to pass through. Hopefully, when we get into the cats and when we talk about vessels, I'll be able to show you, because usually the male cats, you can kind of poke and probe through the inguinal canal. All those structures that serve the testis is called the spermatic cord, and when they pass through the inguinal canal, they create a kind of palpable cord, right? So if you are a male, or you know one very well, you can actually palpate that on the outside of the body. When males develop inguinal hernias, which is very common, You'll have a herniation where the part of the peritoneum is trying to push down through into the scrotum, and that you'll actually have a swelling there, an obvious swelling of that spermatic cord. Basically, the guts are trying to fall into the scrotum. They don't get very far, but that's one of the most common causes of hernia in males. It's extremely common. All right, so here we can see that inguinal canal, the fibers of the dartos wrapping around it, and then the structures of the spermatic cord. You've got blood vessels, obviously. You've got a single tenuous testicular artery. When you see how small this is in the cats and how far it has to go, it'll as a male, it kind of makes you a little bit nervous because it seems rather diminutive. But that's because, remember, the testes develop in the abdomen, and blood vessels branch close to their target. So as the testes descend, the artery has to get longer and longer to serve it. Okay? And then quite commonly, and this is quite common, you'll have a single artery serving a structure and then some sort of plexus or grouping of veins. And this raises the overall diameter of the veins if you add them up together to lower the resistance to drain the structure. And so there's this plexus of veins called the pampiniform plexus. Also within that you'll have autonomic nerves, right? And lymphatic vessels. And of course the vas deferens or ductus deferens, which carries the sperm out of the testis. When a male has a vasectomy, an incision is made into the base of the scrotum, and the surgeon then pulls the spermatic cord out and carefully separates the vas deferens from the rest of these vessels. And then the vas deferens is ligated or cut. Okay? And then everything's put back in. Right? You would not, of course, cut the entire spermatic cord, because that would be equivalent to castration. The testis would die. And not only would it not produce sperm anymore, but it would not produce testosterone, which of course would cause dramatic changes in the male. That being said, this does happen spontaneously sometimes. It's called testicular torsion. The testis will get turned around inside the scrotum, and this will put a kink in the artery and cut off blood supply to the testis. This is usually accompanied by pain, right? I don't have to tell the guys in the room how badly anything happening here hurts, right? So this is what what it feels like when you have this happen. So if you feel like that and it doesn't go away, go to the ER right away, right? Because it may be testicular torsion. You catch it, they can turn the testis back around, and they may even go in and surgically anchor it a little bit they'll induce some scar tissue in those fascia to kind of hold it together a little bit more readily. Okay, but if you don't catch it in time, it easily requires the testis to be removed. Fortunately, one is enough, as with kidneys, one is enough to not only produce enough sperm, but enough testosterone. I have a friend who this happened to in high school. He was playing volleyball. I guess bouncing around the volleyball court caused this to happen. He lost a testicle, and he's got 3 children after that happened. So one is enough. All right, so here we can see a cross section of the testis. We can see, again, that tunica albuginea and the tunica vaginalis with this little space. And that tunica albuginea sends down processes into the center of the testis and divides these are called septum, and this divides the testis into several lobules. And each of those lobules contains between 1 and 3 seminiferous tubules. These then pass towards the middle of the testis and join together in a network, a complex network called the rete testis, which then drains through multiple ducts out of the testis and into this structure known as the epididymis. You can see these individual ducts here coming together, and they're joining this single duct. The single duct is called the epididymis. The epididymis then coils around, becoming very long. I think it's like 20 feet if you were to stretch it all out. Okay? The whole purpose of this is to have the sperm that have been produced in the wall disc, they pass through the rete testis, they pass through the efferent ducts into the epididymis. This gives them time to mature because they need to be exposed to high testosterone levels before they can become viable. Having a very long epididymis acts as a storage mechanism. So the sperm that might be ejaculated will have been sitting in the epididymis for a while. Okay? Now, if they're not ejaculated after a certain amount of time, I'm not exactly sure what the timing is there, they will be reabsorbed. And this is what happens to most of the sperm. They'll be reabsorbed into the wall of the epididymis. And then, of course, the epididymis tapers into that, or becomes that single larger duct called the vas deferens or the ductus deferens. Let's see. Yeah, here's a picture of it from a cadaver. You can see the spermatic cord there with the blood vessels, lymphatic vessels, and so forth. The ductus deferens has been separated. You can see the testis. That white covering is the tunica albuginea. And you can see this kind of comma-shaped structure covering over the top of it. That's the epididymis. And then with the ductus deferens emerging from it. Okay. Now, in between the tissue of the seminiferous tubules within those lobules, you have these cells. They're called Leydig cells. Okay. I think the new name for them is interstitial cells or interstitial endocrinocytes. These are the ones that produce testosterone. Okay. So testosterone is an androgen, a male sex hormone, and it's produced by structures that are outside of the gamete-producing structure. Okay? Put a pin in that in your mind, because we'll see that again with the female. So you have the same sort of thing with the female. Really, I should say, you have the same with the male as with the female, since everyone has an X chromosome. Female is the sort of default development pattern. You need the Y chromosome products to readjust the development pattern to produce a male. Okay. All right. So the vas deferens then rises, passes through the spermatic cord, it enters the body cavity, enters the pelvic cavity. This image shows us a posterior view of the bladder. There's the pubic bone, right? So the pubic symphysis would be right through there. The ductus deferens comes in over the top of the pubic bone, right? It's sort of going up like that because it's following the inguinal canal, and then enters the pelvic cavity. It passes on the posterior side of the bladder, going just over the tops of the ureters. Then it widens, and this is known as the ampulla. Okay? During sexual activity, as the male approaches orgasm, the sperm will rise out of the testes, and that fluid containing the sperm will reach this point. And this is sort of like where it's going to wait before ejaculation occurs. So this is going to fill with sperm produced containing fluid during intercourse. All right, now some other structures we can see. Just near that is a large gland called the seminal vesicle. And this will produce the most of the volume of the semen. I think like 60% of it. In the lecture we'll talk about the different parts of semen and the roles that it plays. Okay? Now notice the duct of the ampulla of the vas deferens and the seminal vesicle join together, producing a common duct called the ejaculatory duct, which then passes through the mass of the prostate gland. The prostate gland sits between the bladder and the pelvic outlet, the muscles of the pelvic outlet, also called the urogenital diaphragm. That's the muscles there, that pelvic outlet. The prostate is sort of a spongy gland. which has multiple ducts that empty into this portion of the urethra. So the urethra passes out of the bladder, passes through the prostate. It's called the prostatic urethra. This is also the location where the ejaculatory ducts enter. You can see their openings there. Right? And this is all before the urogenital diaphragm. So during orgasm, what's going to happen is this external urethral sphincter will relax, and then the other pelvic floor muscles will contract rhythmically. And that will squeeze the ampulla and the seminiferous tubule, and it will squeeze the prostate and force all that fluid out, first through the membranous urethra, which passes through the diaphragm, and then into the spongy urethra, which passes into the penis. Okay? Now there's one more component to semen, and that's added by this gland here. This one sits on the outside of the urogenital diaphragm, so it's not blocked, not requiring those muscle contractions to force it through. This is known as the Cowper's gland or the bulbourethral gland, and it produces somewhere around 10% of the volume of semen. In fact, a good deal of it is released before ejaculation, right? This fluid from the Cowper's gland will travel up the spongy urethra in the penis and then actually leave the body before ejaculation. This is sometimes called a pre-ejaculate. And what it's doing is it's for lubrication of intercourse, and it's also going to neutralize any acidic urine that might still be in the urethra. Okay. If you look at the penis, of course the penis is composed of erectile tissue, and there's two types of erectile tissue here. You'll have the cavernous erectile tissue, which is found in two masses that go along the length of the shaft of the penis. It actually continues into the perineal area where it forms the crus. Then surrounding the urethra and also filling the head of the penis is spongy erectile tissue. And there's a bulb of it near the base, near the transition point between the membranous and the spongy urethra. Those structures there are sometimes called the root of the penis. They anchor the erect penis during intercourse. All right. The cavernous erectile tissue will have a higher pressure in it. It'll be less flexible. The spongy erectile tissue needs to be more flexible so that the sperm can pass, so it doesn't crush the urethra, basically. And then, of course, when males are born, there is additional skin covering the head of the penis, and this will pull back when the penis becomes erect. This is known as the prepuce of the foreskin. Now, it's quite a common practice in the United States to remove that around the time of birth, and whether or not that's necessary and any benefits are something that people debate about. The only thing I know that's it's cleanliness. If you have a foreskin, then you can get this dead skin collecting between the head of the penis and the foreskin, and there's a bacteria that lives on that. It's not exactly pleasant, I suppose, though it is something that humans have dealt with since the beginning of time, so I guess you just clean it like anything else. It also, there's a study in South Africa, and it appears that having a circumcised penis makes you slightly less likely to acquire HIV as a male from a female. So, and probably the biggest potential health benefit is that penile cancer is pretty much always a cancer of the foreskin. So if you don't have one, you're not going to get penile cancer. Very rare. Okay. Now you'll notice these arteries traveling into the erectile tissue, and this is the key to how this happens. During an erection, what's going to happen is you're going to have vasodilation of those arteries. The blood will flow into these cavernous spaces. The
pressure will build up, and that will actually kind of crush the veins that drain the penis, or at least slow down the drainage. And so the erectile tissue is filled with blood, essentially. It's inflated with blood. In order for it to end, the vasoconstriction occurs, and then the blood drains away slowly. Of course, if that continues for too long, you can actually end up with hypoxic damage to that tissue. It's a priapism, a erection that won't go away, and that is a medical emergency. You hear about it in ads for, erectile dysfunction drugs, but there's other things that cause it. And so they'll try to give like sympathetic drugs to try to cause vasoconstriction. If that doesn't work, they just put a needle in there and aspirate the blood out, because otherwise it's going to be bad. All right, let's see, anything else? Oh, again, there's homologous structures, right? So there's homologous cavernous tissue around the vestibule in the female, not quite as much, but it's there. You have the head of the penis is homologous to the spongy tissue of the penis is homologous to the clitoris in the female. So the head of the clitoris is homologous to the head of the penis, and then there is an additional erectile tissue that beneath the head of the clitoris. You have a clitoral hood that's homologous to the foreskin of the male. You have the tissue that makes up the vagina, the lining of the vagina, in the male becomes the spongy urethra. And then there is also a female prostate gland called the Skene's gland that empties a very similar fluid to prostatic fluid into the female urethra. So again, and then there's homologous to the greater vestibular glands, they're called, and they'll produce lubrication for intercourse. They're homologous to the Cowper's glands. All right, so here's a cross section we can see of it. All right, so that's the end of my spiel. More than you ever want to know about that stuff, I'm sure. So we have cabinets over here containing a great many very nice models. So if you look in your lab handout, it'll show you the structures you need to go over.